project is improving cancer immunity by blocking tumor sugar addiction. So first, to understand cancer immunity, we have to understand what the immune system is. So in the immune system, there's two main lines of defense. The first one is called innate immunity. So that's just like a general response the immune system has to any pathogen that enters the body. The second one, if the pathogen gets past innate immunity, is adaptive immunity. So that's when the immune system has like a more specific response to a specific type of pathogen. And so these are the cells of adaptive immunity, and I'm looking specifically at T cells, which are right there. So what is cancer immunity or anti-tumor immunity? So that's basically just the immune system's response to the presence of tumors in the body. So one thing it does is it releases these things called cytokines, which are basically any substance that the immune cells release. And the two really important ones that I'm looking at are CD40 ligand and interferon gamma. So CD40 ligand is a protein that just binds to a receptor on antigen-presenting cells, and what that does is it activates other immune cells like macrophages and B cells, and what those do is they go infiltrate the tumor. And another cytokine is interferon gamma, which pretty much does the same thing as CD40 ligand, shown right here. But one thing it also does is it induces cancer cell senescence. So what that basically is like programmed cell death. So when interferon gamma is released in the tumor environment, then it actually causes the cancer sort of just like shut down by themselves. Another thing I'll be looking at is cell metabolism. So in normal tissue cells, in conditions where there's like enough oxygen, so that's called normoxia, they'll go through this process called oxidative phosphorylation, or OXPHOS, to produce energy. And as you can see right here, it produces around 36 molecules of ATP, which is the units of energy that they use. And it's really efficient, but in conditions where there's not enough oxygen, which is called hypoxia, they'll go through anaerobic glycolysis, which is right here, and as you can see, it produces only around 2 ATP, so it's really inefficient, and it also produces this acid called lactate. However, in cancer cells, it doesn't matter if there's enough oxygen or if there's not enough oxygen, they'll always go through anaerobic glycolysis, which is known as aerobic glycolysis for the cancer cells. So they produce very little ATP, and the majority of the energy they use is turned into the lactic acid. So as you know, tumors they replicate rapidly, so they require a lot of energy if they want to continue growing. And because they use such an inefficient system to produce their energy, they end up using a lot of nutrients in the environment they create. So I did some research last year just looking at the relationship between cancer cell metabolism and anti-tumor immunity. So when we looked at the tumor microenvironment, right here you can see that the amount of glucose in that environment is almost 10 times less than what it normally would be in places where there's no tumors, so like the blood or the spleen. We also found that in conditions where in the tumor microenvironment, T cells actually produce a lot less interferon gamma and CD40 ligand than they normally would in like a tumor-free microenvironment, and so that led us to think that there might be a correlation between the amount of glucose in the tumor microenvironment and the amount of interferon gamma and CD40 ligand that the T cells are producing. So to look at that, I'm going to be manipulating this enzyme called enolase, which is the key enzyme in the glycolytic pathway. So what it does is it basically catalyzes reaction like right about there, and it's actually upregulated in tumor cells, so there's more of it, and so because of that they're able to produce more of ATP for themselves, but they ultimately end up using more nutrients. So what I'll be looking at is if metabolic activities of cancer cells have any impact on infiltrating immune cells in melanoma. So I'm looking specifically at melanoma. And from past research, we hypothesized that suppression of aerobic glycolysis will slow tumor growth and increase effector functions of T cells, so that they'll be able to produce more interferon gamma and CD40 ligand. So to do that, the first thing we're going to have to do is suppress enolase. So we're going to do that by using this thing called a short hairpin RNA, and basically what that is like a short strand of RNA that looks like a hairpin. And what it does is it binds to messenger RNA that codes for enolase, and it suppresses expression of that gene, so it's not turned into a protein that's translated into that enolase enzyme. One thing is that to know it's not the same thing as knockout, sorry. So knockout is when you completely remove the gene, but in knockout, you're just suppressing expression of the gene. The gene is still there. And we're going to be suppressing different regions of the enolase gene. So The two knockouts we have right now are enolase 1 knockdown 1 and enolase 1 knockdown 3. There was a knockdown 2, but that didn't work out, so we didn't use it. After we've made those short hairpin RNAs, we tested the efficiency of those knockdowns. So to do that, we basically inserted the short hairpin RNA into a line of human T cells, and what that does is when the human T cells replicate, they actually replicate with the short hairpin RNA inside them, so we end up with a lot of T cells with a lot of the RNA we need. And after that, we extract the RNA, and then we run a test called a quantitative real-time polymerase chain reaction, which basically looks at the level of that gene expression, so expression of enolase with and without a knockdown vector. So our results, so this enoprimer 1 was our control, so that we looked at as a basis for comparison, and these were our two genes with a knockdown. So as you can see from here, enolase knockdown 1 didn't really knock down that many genes, so it's pretty high. And as you can see from enolase 3 worked really well, so there's very little gene expression. So from that we found that enolase knockdown 3 was the most efficient of the three, and with enolase knockdown 3, we're actually working on it right now, creating, transfecting those into a stable line of melanoma cells, so we'll be able to grow the melanoma, and then after that we will engraft those into mice, and then hopefully analyze the infiltrating immune cells. So for tumor engraftment, after we've generated the stable cell line, we engraft those into wild-type mice, and so on one flank of the mouse, we'll have like a control cell line without the enolase knockdown, and on the second flank, we have the cell line with the enolase knockdown. And so we'll let those grow for around 20 days, and after that, we'll collect the tumors. And once we've collected tumors, we'll analyze the CD40 ligand and interferon gamma production by the infiltrating immune cells. So this is actually what the lab I'm at is going to look at, but I'll be looking specifically at interferon gamma and then CD40 ligand, which is right there. And we'll be doing that using flow cytometry. And so from that, we expect to see increased effector function in infiltrating immune cells, so like more production of CD40 ligand and interferon gamma, as well as an increase in the total number of infiltrating immune cells in the tumor, because if we suppress enolase, then there will be more nutrients in the tumor microenvironment, and so that will give more immune cells the opportunity to infiltrate. So with these results, we hope to get a better understanding of the metabolic competition between tumor cells and immune cells, and through that may lead to a new form of immunotherapy to combat cancer malignancy, such as restoring immunosurveillance by suppressing certain characteristics of tumor cell metabolism. Right. And I'd like to thank my mentor, Dr. Susan Peck, who is the professor of immunobiology at the Yale School of Medicine, as well as Tiancha Guan, who's the PhD candidate who helped me with the majority of this experiment, as well as Mrs. Deborah Day for helping my project didn't exactly go out the way planned, as well as CTJ SHS, and you guys for being an awesome audience.
Great. So Victoria's going to practice fielding questions. So if you have any questions, however simple or complex, she will take them and try to remember to repeat the question, because that's part of the protocol. So we'll help her do that by giving her some questions if you'd like. So I'll act as moderator so I can pick the people who would like to ask questions. Yes.
You said the initial work that you have been doing and what you found so far, that you took the hairpins and actually put them into human cells?
Yeah.
What human, obviously they were cancerous cells?
No, so they're just like normal T cells. So we just put them in there to sort of like grow, so we have a lot of those short hairpin RNAs. Okay,
and did you do work in isolating those? Like, how did you get just human cells? Does Yale Medical just have them, or do you see what I mean?
Yeah, so our lab actually just has like a cell line for those T cells, so we just use those for the knockdown.
Okay.
Remember to repeat the question.
Okay.
I had like 2 half questions.
That's okay. Plus it gives you a couple minutes to just kind of like collect your thoughts too. Other questions?
You'd mentioned that there were 3 different types originally, and you said
Yeah.
the second did not work.
Yeah.
Was that something that was tested, and then you decided to not even bring it forward in your study, or?
So just like repeat the question. The question was, I had a second knockdown, but it didn't work, so I just not decided to bring it up. So it actually happened when we were like making it. Something happened when we were making it, so it actually ended up not working. So we're like, we might as well just not use it at all.
Okay, so it wasn't a viable sample or something.
Yeah.
Thank you.
What specifically did you do in your project?
It's so awkward repeating the question. So the question was, what specifically did I do in my project? So so the short hairpin RNA is actually already made. So I actually ended up just, hold on, I gotta look back at
Yes,
this.
that's fine.
Alright, so here, I did the transfection and I also extracted them from the cells and ran the PCR to test efficiency.
And then last year's study also.
Yeah.
Even though this wasn't a compounded study, you did a lot of base work to get to know your skills and techniques and things like that too. Did you have a question? I'm sorry to cut you off.
Yeah. What materials did you need to execute this procedure?
So the question is, what materials did I need to execute this procedure? Well, we needed T cells. We also needed just like a qPCR machine, and they also like a kit to extract the mRNA. That's basically it.
What were some of the obstacles you had to overcome?
Okay, so the question was, what were some of the obstacles I had to overcome? There was the initial one where we were going to use a knockdown that was inducible, so it wouldn't be activated until there was a chemical that activated it. But that knockdown actually didn't end up working, so we had to switch to a normal one so that wasn't inducible. There was also the time problem where my mentor was in China for 5 weeks, so that set back a little.
The original projection was
Yeah.
that you'd be completed by mid-March, but these things happen.
Yeah.
So that's why you're still working on it. So you'll have some more results for by the time you actually are presenting at JSHS in 2 weeks. So that's
Yeah.
good news too. So, but you're most of the way through. So great effort. Anybody else have questions? Excellent work. And Katie's next. I have to stand on this side. Or you stand wherever you're comfortable. Do you want me to?
Yeah,
advance it for
could
you?
you?
I just can't give you any feedback at the same time.
Or I'll do it.
I think
So
I've
I'll
given
do
you
it.
enough.
I'll do
You
it.
want to do it?
I'll be honored, Katie.
I don't know. I like this side better for some reason. You
Oh,
do.
sorry.
I think it's the party hat.
Oh, thank you.
Okay. Hi, my name is Catherine Handler, and my project is Succession Variations in Kenyan Scavenger Communities and the Importance of Wildebeest Carcasses. So to begin, there's complex relationships between aquatic and terrestrial ecosystems. My project focuses on the Mara-Serengeti ecosystem, which you can see in this map here is located in Kenya. And in this ecosystem, this is an ideal location to study the complex relationships because there is the Mara River, which runs through it, as well as the surrounding grasslands. So wildebeest are a primary species in this area. Approximately 7,000 wildebeest drown annually in what are known as mass drowning events because in seasonal migrations, they have to cross the Mara River. However, sometimes the current is too strong or it's especially deep that time of the year, so they cannot cross it and they end up drowning. This leads to a huge input of carcasses into the river, which are very rich in nutrients. So what's happening to these carcasses also asks what's happening to the nutrients in these carcasses. So in a study on Peru vultures conducted by Wallace et al. In 1987, found that the more dominant a species was in the scavenger guild, the more access they had to the carcasses, and a species was more dominant, the greater body mass they had. And then since my project is in Africa,
Some African studies found that there is a positive correlation between small vultures and large vultures, where if one is present, the other is likely to be present as well. And in addition, the habitat use of the scavenger species correlates with the abundance at these carcass sites. So Kendall et al. Found that the Rappel's griffon vulture, which is pictured here, was less abundant in the scavenger community than other species due to the fact that its nests were in cliff dwellings, which are significantly farther away from the carcass sites than other species. And this study also found that the carcass size correlated with the number of scavengers at that carcass. And so last year I did a study where I analyzed a photo series taken by my mentors on an automated game camera. I'll get more into the details later, from November 2012. And my two significant conclusions were that there was a decrease in the number of scavengers around day 12. And around this time, there was also a change in the composition of the scavenger community, where the larger body mass scavengers, such as the marabou stork, wetback vulture, and Rappel's griffon vulture, became less abundant, and the smaller bodied scavengers, such as the sacred ibis and the hooded vulture, became more abundant. And so the purpose of my project this year is to validate those previous results by analyzing an additional photo series taken in October 2013. And this involves asking what scavengers are eating the wildebeest carcasses and what temporal trends are evident from days since the drowning event. And my second objective is to analyze the importance of these carcasses to the scavengers. This involves asking what are the metabolic rates for the scavenger species and how much of the available DM, which stands for dry mass of the carcasses, is being consumed by the scavengers. And my third objective overall is to see how the nutrients are rotating between the aquatic and terrestrial ecosystems in those complex relationships I mentioned earlier. And so my independent variables are the time from the drowning event measured in days, the time of day, and the photo series. So I mentioned I have one from November 2012 and a second taken in October 2013. And then my dependent variables are the different species that are present at these carcass sites and the number of individuals of each species. And so my first hypothesis is that the temporal changes viewed in the previous study will be observed in the new photo series, as well as the scavengers consume a large proportion of the carcasses and thus are important in the nutrient transport from carcass decomposition. And so these are a brief summary of my materials. I'll get more into that in my procedure. However, the photos were taken by a game camera set up by my mentors, and then I have
the following teachers please report to the main office. Maggie Barkin, Ashley Karan, Peter Downhour, Dave Pepitone, Eddie Anderson, Greg Tuhill, Joyce Narden. Thank you.
Okay, and so a study by Komen in 1992 supplied estimates for metabolic equations. And so there are three main parts of my procedure. There's the data collection, data analysis, and metabolic modeling. And so for the data collection, next slide. So the automated game camera was set up during field research by my mentor, Amanda Suboleski. And this camera took pictures approximately every 15 minutes. However, I only analyzed the common avian scavengers once every hour on the hour. However, I did end up going through every photo because for rare observations such as non-avian scavengers or camera movement, etc., I looked through every 15-minute picture. So I ended up analyzing 1,273 photos from the November 2012 series last year. And this year, I analyzed 1,802 photos from the October 2013 series. And so if you go to the next slide, you can see two examples of a photo that I would look through. So in order to analyze this photo, you can see the camera records the temperature here, and then I go through and say what species are here. So you can see a marabou stork is flying here, and there's another one here at the carcass. So I would say what species are here, what number of each species are there, and then how many are consuming the carcasses. And I define consuming the carcasses as being on or in close proximity to the carcasses, and having their head close down or bent down, so like eating the carcasses. And so for the metabolic modeling, these estimates are all from a paper by Komen in 1992. EE stands for energy expenditure. I multiplied a coefficient supplied from the literature, 826.7. I multiplied that by the body mass of the species of avian scavenger raised to the exponent 0.61. And then this, so the energy expenditure is how much the...
Brendan Moss, please come to the main office. Brendan Moss to the main office.
So the energy expenditure is how much a species needs to live, And then in order to see how much energy they actually need to consume, we took into account the energy assimilation efficiency, assimilation coefficient, which is 0.86, to see how much is actually going away in heat, so to see how much they actually need to consume in order to survive. And then in order to find out how much, then I calculated the dry mass consumed by the species, and so this was divided by 26.0, which is the energy content of a carcass, to see how much they can get from these carcasses. And I multiplied this number by the daily maximum number of individuals of the species per day. And so this graph shows the stacked averages of common avian scavengers from October 2013 series. The avian scavengers were the most common scavengers overall, from like terrestrial and aquatic scavengers. And however, you can see that the marabou stork is the most abundant in the species, in the scavenger community. And if you look in this, the percentages graph, you can see that the composition does change again, where the sacred ibis, which is a smaller avian scavenger, becomes more abundant, and the larger scavengers become less abundant. This occurs at around day 17. However, if you remember from the background, this occurred at day 12 in the November 2012 series. So I asked, why was this happening so much later in this series? So to answer that question, I compared the two photo series, and I found that in the November 2012 series, there was almost a quarter of the number of carcasses from the actual drowning event, which means that in October 2013, there's 4 times as many carcasses available to these scavengers. And then in the actual carcass aggregation that I analyzed in the photo series, the October 2013 was more than twice the size of the November 2012 series. So what this means is that if you consider or guess that there's roughly the same amount of scavengers in the area, they're going to be more spread out in the October 2013 series due to the huge influx of resources. And so to further test this, I looked at how much, this is where the metabolic modeling comes in, and I looked at how much the scavengers were actually consuming. So this is a comparison between 2012 and 2013, and the total percentage that was consumed by the 5 common avian scavengers in 2012 was 12.9% of the DM available in the carcasses in November 2012, and 6.91% in October 2013. So this just supports the conclusion that because there are more carcasses available in October 2013, the trend occurred later because the rate of decomposition will be slower because there's so much more resources available. So to get to the same level where the larger scavengers are able to be less abundant and the smaller scavengers are more abundant, it is going to take longer and they're consuming less of the carcasses there. And so the first hypothesis was partially supported. There were similar trends, however, as mentioned, occurred later. And then my second hypothesis was supported that the scavengers play an important role in the transport of nutrients. This is due to the fact that they are consuming a large proportion of the carcasses there, and they're spending all of their hours of every day in this time eating these carcasses at these carcass sites. So they're very important in the transport of nutrients. And due to the fact that they're only consuming roughly 6 to 12% of the carcasses, that shows that they're stopping where they need to stop because there's still a lot more carcass available to them. So because they're stopping where they need to stop, from that we can conclude that carcasses are a bountiful resource to fulfill energy requirements. And so there are several limitations of this study. It was a photo analysis study, so that means that I was limited to see what was actually in the frame of the photos. And in addition, the light exposure could have slightly altered the identification of these species. However, my procedure accounted for this because if I was not completely confident in what species I was looking at, then I counted it as unknown for the species. However, this means that the estimates in this study are underestimates because the unknown numbers, the individuals, were not accounted for in the calculations for the metabolic modeling.
10 minutes.
And the number of days in this photo series are also a limitation. The November 2012 series started 5 days after the drowning event, and the October 2013 series started 4 days after the drowning event. So for the metabolic modeling, we made an assumption that for days 1 and 2 after the drowning event, based off of a personal observation by a separate researcher, we assume that there are 0 vultures at the carcasses there. And then for the days 3 and 4 that were not accounted for in the photo series, we assumed just an average for the other days. And so the implications of this project, it shows that the importance of wildebeest carcasses, they are very important to these avian scavengers. And the three vultures that are in this study, the hooded vulture, the white-backed vulture, and the Rappel's griffin vulture, are all endangered species, so their numbers are declining. And since these carcasses are a resource for them to fulfill their energy requirements, these are very important in their lifetime. And this also shows what happens when an organism dies, what scavengers are consuming the carcasses, as well as the recycling of nutrients. So this study focused more on what scavengers are consuming them. And so in the future, we're collaborating with a researcher, Corinne Kendall, who has radio telemetry data on the vultures in this area to show the movement of these vultures and thus the movement of the nutrients between the aquatic and terrestrial ecosystems. And I'd like to thank my mentors, Dr. David Post and Amanda Suboleski at Yale University, and Ms. Deborah Day at Amity Regional High School, as well as JSHS for allowing me to present here today. Thank you.
25 seconds to spare. So, well done, Katie.
Thank you.
Do we have questions?
What is a temporal trend?
So the question was, what is a temporal trend? And temporal trend refers to the changes in the scavenger community over the course of the days since the drowning event. So I showed the graph where the stacked averages of the avian scavengers. That shows the temporal trends of the marabou. The numbers all were increasing from days 4 to around 14, and then they started decreasing. So that's an example of a temporal trend.
So what do you see as future off of your study or from this point on for what do you expect to get from this data or use with this data for? If that's the best way.
So the question was, what do I expect to do in the future with this data and see from this data? So, well, first I mentioned that we were adding the radio telemetry data to add the aspect of the movement of the vultures to show the recycling of nutrients between the aquatic and terrestrial ecosystems. And then we're going to most likely analyze another photo series just to have 3 to further validate the results. And then off of that, we can show the importance of these carcasses and possibly lead to conservation of the avian scavengers or the wildebeest.
For the camera, all the photos that you analyzed, was there one picture taken from one camera at a certain time during the day, or were there several locations of cameras set up?
Yeah, can you go to the background with the map? So the question was there one camera looking at these photos or several around the area? And so, yeah. And so this map shows that there's 3 sites, and these are for 3 common crossings of the river. And so my mentors kind of set these cameras up as a whim. It was very spontaneous. They were just curious to see what was actually going on. So for each carcass site, there's only one camera looking at it, so I only have one angle of it. And the 2 series that I looked at are both from around Site 3. However, they're mainly, they're in Kenya from 2009 till today. And so they have multiple series from around this area from different cameras.
The second series of photos that you analyzed, were they put in specifically the same Site 1, 2, 3 or no?
The question was, were they specifically put in Site 1, 2, 3? And no, they were not. They're 100 meters away from each other just because that was where the carcass aggregation was.
Okay.
And however, these are the sites just refer to where the wildebeest actually go to cross the river.
Okay.
This is a question from a non-science person. I'm kind of I'm just really impressed and blown away by the research that you're doing. You talk about the different vultures that are endangered species, but I'm looking at all these wildebeest that are drowning annually, and how is that population? Is that the wildebeest population? Is that a flourishing population?
Yeah, currently is. I have the number of total wildebeest in the area, and I don't have it off the top of my head. The main reason I point out that these are endangered species is because wildebeest are ungulate species, so they're both hoofed, and that puts them in the same category as bison in America. And so bison numbers have been decreasing over the years, and so since they're ungulates, we can hypothesize or suspect that their numbers will start to decrease as well due to their similarities. So if this does happen, then we can see how if the because the carcasses are a resource for the endangered species. If the wildebeest then decrease, this will show how it can affect the already endangered species.
So the wildebeest at this point are not endangered species?
Correct, yes.
Okay, thanks.
So, and this might be a follow-up to yours in terms of the future thinking piece.
Mhm.
I see a lot of this looking back and trying to find out what's affected the vulture population. Is there something that moves forward that would be a man-made change that would affect either one of those populations? Is there a purpose in that respect?
So the question was, is there a purpose for looking at this based off of man-made factors affecting the species that are studied in this study? And so currently, there is no really important human interference with this. However, as we can see for other species, there is a lot of poaching going on, illegal poaching. So although this might not pertain entirely to this study, it could be affected in the future, and then we would know that this is an important area to conserve.
Good job. Thank you.
Scott DeMayo, please call extension 3510. Scott DeMayo, 3510.
You can grab here, Victoria, we can give you your feedback right now. That way, if anybody has feedback for
when I
Victoria,
say
she's
it,
gonna get to the lab. Okay, sure, we can do, yep.
Where the PE
Can I ask you a logistic question?
I
What
could
am I
like
supposed to be
change
putting in
that.
the spaces?
Oh, you know what, it's
Yeah.
just, this is the only thing I give for a checkbox, kind of that the judges they're going to check for. So we don't,
Or I don't.
just, are they there, or were they present?
Yeah.
That's all basically.
Yeah,
Because well, we don't know what the judges do with them or what their scaling system is, they just give us that as an outline, sort of this, scaffolding. So- yeah. Or if something was unclear or too complicated, maybe you can just write a little note. Yeah. Great.
I apologize, my feedback limited.
No, no,
I
it's
called
fine.
out the SAT there, so.
Thank you.
Great. She's gotta get to the lab, you know. Thanks, Victoria.
Bye.
Bye.
Finally.
And without further ado, we know Helen Liu.
All right. Hello, everyone. My name is Helen Liu, and my project is developing a novel biologic for Gaucher disease. Gaucher disease is the most commonly lysosomal storage disease, and lysosomal storage diseases are a group of diseases in which a mutation in their enzyme has occurred. So in this case, for Gaucher disease, the enzyme that's mutated is glucocerebrosidase, GBA. What happens is that patients suffer a deficiency of GBA in their lysosome. The function of GBA is to digest its substrate, but because there are reduced levels of GBA, this causes the accumulation of its substrate beta-glucoceramide. The aggregation of beta-glucoceramide causes enlarged lysosomes, as you can see, and then causes the enlargement of several organs, most known for its spleens, livers, and lymph nodes. In addition, patients also suffer anemia, which is blood weakening, and osteoporosis for their bones. So the currently most used treatment for Gaucher disease is this enzyme replacement therapy. Enzyme replacement therapy works by the patients receiving an IV of this deficient GBA enzyme. However, there are several problems with enzyme replacement therapy. The first is that it's extremely expensive. Enzyme replacement therapy costs on average $350,000 per patient annually. In addition, there have been cases of immune response or adverse effects reported. So what this means is that not only did some people not find the treatment effective, but others even found more harmful effects. In addition, since enzyme replacement therapy is the only currently used treatment, there is an extreme need for an alternative treatment. Progranulin, which is what I worked with, is a specialized protein known as a growth factor. Literature research has found it to be involved in numerous functions, including inflammation, wound healing, infection, tumor genesis, degenerative diseases, and only recently to the lysosome. So in this first study done by Tanaka et al., what it most importantly showed was that there was a key connection between progranulin, this protein, and the lysosomes and its functions. The second two studies, Goetzel et al. And Smith et al., what they showed is that for Goetzel et al., they used a mice that had deficiency of progranulin. Smith et al. Used cells that do not have progranulin. And what they both found out was that these cells or mice contracted an LSD known as lipofuscinosis. And so what this led us to ask is the reverse. Can we use and determine whether progranulin is therapeutic in LSDs? So can we reverse the process? We know that progranulin, the loss of it, causes an LSD, so can we treat an LSD with this program? So, if we can find that there is indeed remedial effects, can we develop a drug based on this knowledge? In our first study, we preliminarily tested 7 different fibroblasts from 7 different lysosomal storage diseases. So this first approach is the lysotracker approach. What happens is we have these fibroblasts, these cells, and what we do is we add lysotracker dye. So lysotracker dye, how it works is it stains lysosomes with a red color. And this is not actually just one lysosome. Instead, each mini little dot constitutes a lysosome. But the more lysosomes you have, or the larger the lysosome, the more red color you have, and thus the more intense the red color is. So we can see in the normal, there's no color because the lysosomes are spread out. But in the lysosomal storage diseases, there's an intense red color. But when we treated or added our recombinant progranulin to the fibroblasts, we saw that the Gaucher disease fibroblasts were successfully reverted back to levels that were similar to the normal cell type. So what this led us to test is whether recombinant progranulin truly prevented GBA aggregation and beta-glucoceramide accumulation. So whether adding progranulin was involved in the processes that caused Gaucher disease. So this study is called, or this experiment is immunofluorescence staining. So this meant that we chose a color for each item that we want to measure. So we stained GBA levels with green, beta-glucoceramide with red, and then nucleus for blue, just as a control and the localization purposes. So you can see in the control, the levels were very normal, that there's hardly any color that you can see. Next, we added a lipid mixture. So what this does is it stresses the Gaucher disease fibroblasts. It causes more GBA to be produced and more beta-glucoceramide. That way, when we treat it or add progranulin in the final step, we can see more significant effects. So you can see after the lipid treatment, it successfully upregulated the GBA and beta-glucoceramide levels. But after adding recombinant progranulin, it, again, like the previous image, reverted back to normal control levels. Next, we want to analyze whether progranulin was involved in the lysosomal appearance. So whether it was involved in bringing GBA to the lysosome, which is the important part that causes Gaucher disease. So in order to do this, we used an siRNA approach, which meant we targeted knockdown efficiency. So what we did is we transfected our cells with an siRNA, this targeting progranulin. So what this does is it targets progranulin levels to be knocked down, the gene expression to be lower. After that, using this probe, the MDW933 probe, what this does is it measures GBA levels. So using our knockdown of progranulin, we also saw that there were undetectable levels of GBA as well. So what this meant was that progranulin was essential for the lysosomal appearance, that in order to have GBA in the lysosome, you needed progranulin. Afterwards, we wanted to see how these two interacted with each other. So in our first experiment on the left, we conducted a co-immunoprecipitation assay. So how this works is that we transfect cells with, but add markers to what we want to measure. So in this case, we tag progranulin with flag tag and a GBA with the RFP marker. Next, we incubate these two together, these cells, and we use the antibody of one of them, in this case progranulin, and we cause it to basically isolate progranulin alone. And whether if progranulin did bind to GBA, when we use the antibody to detect GBA levels from this isolated progranulin, it should be present. So that's why you see the band here. And what this band means is that yes, indeed, there was binding between GBA and progranulin. So next we want to determine whether this was through indirect or direct binding. So we conducted a solid phase binding assay. In this assay, we only use purified substances. So we use GBA and 2 other transport proteins known that interacted with GBA. What we saw was there was only binding activity to this GBA molecule, that progranulin did not utilize or bind to any of the other transport proteins. So this meant that progranulin directly binded to GBA. In terms of drug development, it is better to have the smaller molecule. First of all, because it's much cheaper or easier to produce smaller molecules, which thus reduces the cost. In addition, especially for the case of progranulin, which is a growth factor, increased progranulin can increase your risk for cancer. So thus, we only needed to find this essential fragment, this small piece that still retained the function that we needed. So what we did, the lab had already created several different constructs of progranulin mutants. So what I did afterwards was I took these constructs and, again using co-immune precipitation, I saw whether there was any binding activity to GBA. And we found that only the full-length fragment retained this binding activity. So that meant somewhere in the granulin E region was the necessary part. What we did then was we conducted another co-immune precipitation assay, but using first the granulin E region. Unfortunately, we saw no binding activity, so we extended a linker region here. And luckily, we found that there was indeed a band of binding activity. So we nicknamed this PC-GIN, which is progranulin C-terminus for GBA interaction. What this means is just this PC-GIN molecule is the essential fragment in order to bind to GBA. So of course, we have to see whether PC-GIN can actually treat it like progranulin did. In this first experiment, which is transfection, what we did is we, again, used the lysotracker approach. So we stained our lysosomes with this red color. Next, we transfected our fibroblasts, but added a GFP or a PC-GIN GFP fusion. So what this protein does is it allows us, when we turn on fluorescence, to see a green color. So afterwards, because what we should see when we merge the 2 images is that we should ideally see the green color retained, because the lysosome levels should return to normal. We shouldn't see all this red color that we did in the beginning. So we saw that indeed, when PC-GIN was added as a treatment, it retained its therapeutic effects.
10 minutes.
Thank you. So next, in order to be viable or applicable in actual drug treatment, the body will take it up through as a protein. Unfortunately, we cannot transfect humans. So what we next conducted was adding recombinant progranulin. So we took our fibroblasts, and instead of transfecting cells with PC-GIN, we simply added the protein. And yes, indeed, we saw that they reverted to normal, similar to the control, affirming that PC-GIN had therapeutic effects. So why PC-GIN ideally works is this is our hypothesis. So PC-GIN is an 88 amino acid length. What its 3 most important components are its binding motifs to GBA, of course, and to HSP70 and sortilin. HSP70 is this specialized protein which is involved in trafficking. So what this means is it can direct an item or a molecule to wherever it should be in the side of the cell. So HSP70 is responsible for directing PC-GIN or progranulin to the lysosome. Sortilin is a receptor, a lysosomal receptor. What this allows is the endocytosis, so for progranulin to be taken in by the cell, so that's why it can be a viable drug treatment. So our most important parts, we found that progranulin directly binds to GBA, and it could actually transport this deficient GBA back to the lysosome in order to remediate the effects of Gaucher disease. And we found the necessary C-terminal fragment of progranulin, which could be a viable drug treatment. So this graphical summary summarizes our results. The proteins, progranulin and GBA, will be created in the ER or your ribosome, next transported to the Golgi. Afterwards, progranulin will bind to GBA and create this complex. It will also bind to HSP70, and HSP70 will direct it to the lysosome. It will also bind to sortilin, which allows for endocytosis, so that's why it's in the endosome. And then, after it separates, it can successfully transport the GBA enzyme to the lysosome. So what this means is progranulin is this indispensable piece to our puzzle of how we can cure or treat Gaucher disease. So in future research, we obviously need to test Gaucher disease in preclinical animal models. We need to evaluate pharmacokinetics, stability, and toxicity of PCGen, properties that we normally control. Also, because we only conducted a study with 7 different lysosomal storage diseases, there are about hundreds of them. So we could examine whether PCGen could be applied to other lysosomal storage diseases. And I would like to thank my mentor, Dr. Jinlong Jian, everybody at the lab, and all the wonderful people in my journey. And also thank you guys for being an amazing audience.
14 minutes.
Oh,
We'll shoot that up to 15. We're not exactly sure how
Yeah.
long, if they're just going to keep you in conversation or what behind the scenes there. So you may be interrupted and be asked questions throughout. So Helen can get used to kind of fielding everything, because the science fair experience actually has helped you out a lot with that. So, yeah. Questions?
So you did the 7 cells that you did the testing with, the
oh, the
staining,
first one, yes.
So the first 2 were Gaucher.
Yes.
What
Oh, I
were-
can go through them.
What were the other 5 for? Those, right there.
Yes. So all 5 of these are lysosomal storage diseases. There are about hundreds of them that I've said. So this is Niemann-Pick disease, Fabry disease, muco, I forgot what the L stands for, and I forgot the names of those 2 off the top of my head. But these are all different ones. So they all have a different mutation, except for Gaucher disease. Gaucher disease actually has 3 types, which range in severity, type 1 being the least severe, but the most common, like 95%. So we mainly focused on Gaucher disease 1.
I've never heard of Gaucher disease or Gaucher or however you
Yes.
pronounce it. What made you choose this?
So it is a relatively rare disease, that like 1 in 40,000 people, so that makes sense why you haven't heard of it. I didn't actually start off with Gaucher disease. I wanted to do arthritis research. So I contacted a lab that was doing arthritis research, but was also doing progranulin. And so what happened was I was reading papers on progranulin. I'm pretty sure it was, I think it was this one, and there's also an even earlier study done in 2010, pretty much saying the same thing, that the progranulin loss of it caused an LSD. So it's fairly simple since we had this recombinant progranulin already, and the lab just needed to purchase a couple fibroblasts or cells. So my mentor was like, why not? We can make this a side project. And then it was like, yay, it worked. So very, so very exciting.
Going back to the previous slide you were just on, I'm just curious, did you choose and I'm going to say it wrong, so I apologize, Gaucher disease?
Oh no, it has multiple pronunciations.
I'm sure. Did you choose that because that was the only one that seemed to show the best results to the progranulin? Or was that, you started with that and then said, well, let's also look at these other ones simultaneously? Where was the starting point?
So the starting point is we chose these 7 because they're the most common lysosomal storage diseases. And then this spearheaded, Gaucher disease, it looks pretty good, let's keep researching it. So.
Probably a lot had to do with the resources at your lab,
Yeah,
what you
yes.
already had
Yeah.
available to you as a starting point.
Definitely.
And obviously, this is not a 2-month or 3-month process.
Oh.
Helen, how long have you been working on this?
I started freshman summer and I went back on some day we had off to get my last experiment on here.
So all the way up, but mostly during the summer?
Yeah,
period?
definitely mostly things.
So freshman to sophomore, so it's 3 years
3
during
years.
the summer,
Yeah.
almost every day in the lab?
Well, the first year it was like learning techniques, so it was only like 2 days a week.
Mhm.
But then it, like the next summer, it's more regular.
Yeah. And so for your future work, what's a projected amount of time that would take in order to kind of
Oh,
bring this full circle?
well, clinical trials take like 10 years. So realistically, it's not going to happen very soon. But I believe.
I hope so.
What is the cost comparison between this treatment, because you had mentioned the
Mhm.
other one's so exorbitantly expensive?
Yeah.
Is this a less expensive option?
Definitely. So the reason why the other one's so expensive is it's the entire enzyme. So it's like an entire protein, whereas PC-Gin is only this like 88 amino acid length. So it should be.
Easier to crank them out.
Yeah.
Okay.
Thank you.
What's the best way to isolate that fragment?
Oh, to find PC-Gin?
To-
Like how we determine the deletion mutants?
Well, once you determined it, but then if you wanted to extract it later from the larger enzyme, is it already isolated, or would you have to go through an extracting technique in order to break it down and make it the correct molecular sequence and things like that?
Wait, extracting the enzyme, what do you mean by that?
I thought you said it was from a larger molecule to begin with, so I guess you were
Oh, like the GBA treatment?
yeah, like the GBH. So it would
Oh.
be totally different than that.
Yeah, no, I think they're pretty much using like the entire enzyme.
Yeah, I didn't know if you had to start with larger molecular entities and then break them down and do that chemically or treat them somehow.
Yeah. I'm pretty sure there's definitely research into like, there's several enzyme replacement therapies all based on GBA. So I'm pretty sure they differ from each other somehow. So probably should try and find the shortest.
What are you most excited about going to Intel?
Oh, the food in Obama.
His wife's there, she's not gonna let you eat anything good, so.
Oh.
That's coming up the week of March 10th. Is that when
So
you
that's
fly
next
out?
Wednesday.
Next Wednesday.
Oh, next Thursday.
Next
Alright.
Thursday you fly out, and it's all expense paid, and there's where are you staying?
I have no idea.
You just show up at the airport and they have a big sign that they hold, and then you
What city?
DC.
Oh,
Yeah.
okay.
Yeah, and then every day she'll have some sort of judging for a couple of days, and then there's a public event on Sunday. And the people judging her, a good number of them are Nobel laureates or very prominent science researchers. No problem, no
No,
pressure.
no,
We've
it's
done-
fine. No, it's fine.
So she's ready for it, right?
I hope. Well, just
There-
the reminder that you gave to the others that you didn't give to Helen was to repeat the question.
Oh, because mine is going to be different since I'm doing a poster thing, so it'll be like everyone's crowded together.
I see. Okay,
Under
so,
poster,
yeah, makes
yeah.
sense then.
Now you have to make 3 different posters for this, right? There was-
Only 2.
So only 2, right?
Two.
Okay. Because you use one for 2 different purposes.
Yeah.
Right? So you'll have one for the event that's the public day. That's the really kind of big one, and then a smaller version of that for the gala night that you do sort of during the cocktail hour. I guess you just have a social.
Yeah. Listen to my research.
Very exciting.
Good.
Thank you.
Thank you, everyone, for
Well, I'm glad
coming.
because I
Thank
had a
you.
meeting that got canceled, so I
Oh,
had it on my calendar, and
wonderful.
I always try to